Good morning or good afternoon, depending on which part of the world you're joining us from, and best wishes to all of you on International Pathology Day. A warm welcome to this webinar hosted by IRA Matrix and Experimental Pathology Laboratories Incorporated. My name is Dr. Uttara Joshi. I head business development at IRA Matrix, and I'll be your moderator for this session. We have a fantastic talk coming up titled Application of Artificial Intelligence in Grading Rodent Cardiomyopathy. I'm just going to introduce our two expert speakers for this talk. The first speaker today is Dr. Deb Tokarz, and then she'll be followed by Mr. Nitin Singhal. Dr. Deb Tokarz is a toxicology pathologist at Experimental Pathology Laboratories based in Durham, North Carolina, where she provides pathology support for commercial and academic toxicology studies and the National Toxicology Program. Deb completed her veterinary training at Michigan State University and a residency in anatomic pathology at the University of California, Davis. She has a PhD in comparative biomedical sciences from North Carolina State University. Deb has been integral in the development of digital pathology and artificial intelligence applications at EPL. She will be speaking today about the rationale behind the solution, the development, its application, and the results. Our second speaker is Nitin Singhal. Nitin is a technology professional with a track record of developing a repertoire of innovative AI-powered imaging applications. Nitin heads product development at IRA Matrix and will be elucidating on the technology behind the solution. A few housekeeping tips before we begin. All attendees will be muted during the event to ensure quality of the audio. If you have any questions, please type them in the Q&A box and we will be happy to discuss them at the end of the webinar. An email will be sent to all registrants with a link to the on-demand webinar within 24 hours after this talk concludes. So let me begin with a brief introduction to IRA Matrix. So sorry about that delay. Yeah, we are a technology company that provides image analysis and image management products and services for toxicology, pathology, and discovery pathology, among other domains. IRA Matrix was founded in 2011, and we've been working in histopathology image analysis for close to a decade now. We are a team of 60 professionals with expertise in different areas like toxicologic pathology, oncopathology, machine and deep learning, data analytics, high performance computing, and productization. We work in collaboration with pharma companies, CROs, and oncology research institutes across the globe on an array of image analysis solutions. We develop AI-based image analysis and image management products and solutions for toxicologic pathology, discovery pathology, oncopathology, ophthalmology, and microbiology. And we cover practically the entire gamut of applications from screening and triaging to disease quantification to disease grading, as well as prediction. So we aim at moving beyond the conventional digitization paradigm of machine and deep learning to harness their true potential with applications aimed at automation, operational efficiency, prognostication, prediction, and precision medicine. We work on a platform-based services model. Sorry, I'm having a bit of a problem with moving my slides ahead. Please bear with me. Yeah. We work on a platform-based services model. What that means is We provide solutions for pathology image analysis using Airavat, our AI-based platform. This is to ensure that we do expedited development of custom solutions and efficient deployment of off-the-shelf products, in addition to other obvious advantages of a platform-based model. Our solutions are essentially aimed at serving three goals: automating manual processes, improving the efficiency of laboratory workflows, and drawing predictions from analyzed data. In the automation area, we have worked on solutions for detection and quantification of hepatic steatosis, hepatic fibrosis, glomerulosclerosis, and different nuclear and membrane biomarker quantifications. On the workflow efficiency side are solutions like tissue triaging systems for toxicology pathology, automated staging systems to aid multiparametric evaluations in reproductive toxicology, solutions for quantification of acute and chronic lung injury models, among others. In the predictive analytics domain, we concentrate essentially on predictive toxicology, precision diagnostics, and microbiology. We work with pharmaceutical companies and CROs across the globe to develop AI-based image analysis solutions customized to their specific needs. The collaborative model ensures that we correctly understand and translate client requirements to productized solutions, also providing deployment options commensurate with client infrastructure. And the talk today is about one such exciting collaborative project between EPL and Aira Matrix for the development of a deep learning based image analysis solution to score severity of rodent cardiomyopathy. And with this, I'll now hand it over to Deb to talk more about the solution, to be followed by Nitin, who will talk more about the technology.
All right. Thank you, Uttara, for that introduction. I'm very excited to talk today about this project in which EPL partnered with Aira Matrix to develop an artificial intelligence-assisted algorithm for detecting and quantifying cardiomyopathy in rat heart sections. So many of you will be familiar with rodent progressive cardiomyopathy, which I will refer to as PCM. This is a common spontaneous microscopic finding in rats and mice that increases in incidence and severity with age. In general, PCM is more common in rats than mice and occurs with greater incidence and severity in males than in females. PCM is characterized by a constellation of microscopic changes. It can first be recognized as foci of cardiomyocyte necrosis, accompanied by inflammatory cell infiltrate that is predominantly lymphocytes and macrophages. This progresses to varying degrees of fibrosis in later stages, and in addition, mineralization may also be present. Importantly, cardiotoxic agents can increase the incidence and severity of PCM or cause microscopic changes that overlap with PCM, such as cardiomyocyte necrosis and fibrosis. So in order to detect test article-related cardiotoxicity on this background of spontaneous PCM, pathologists must approach diagnosis and grading of PCM in a sensitive and consistent manner. And this can be a challenge given the spectrum of microscopic features, as well as temporal changes that encompass PCM. We hypothesized that this challenge could be met by computer-assisted image analysis. So EPL approached Aira Matrix with the goal of developing an AI-assisted algorithm that could be used to evaluate a histologic section of rat heart and detect the individual microscopic features of PCM, including cardiomyocyte degeneration and necrosis, mononuclear cell infiltrate, fibrosis, and mineralization, and then to provide quantitative output in terms of the percentage of the heart affected by each of these features. We obtained cases for training, validating, and testing the algorithm from the National Toxicology Program archives. The archives were searched for rats with the diagnosis of cardiomyopathy, and then hearts with a diagnosis of no abnormalities were also included to make up approximately 10% of the cases that we pulled. We included Harlan Sprague-Dawley rats from both sexes and from subchronic as well as chronic studies. During training and validation of the algorithm, samples from both control and treated animals were used. For the testing set, only control rats were used. Hearts that had a significant concurrent diagnosis, such as a neoplasm, were excluded so that this initial project could be focused on detecting PCM-related features. H&E stain slides that fit the selection criteria were obtained from the archives, checked for quality, and then scanned to whole slide images, or WSI, using either an Apereo or a Hamamatsu scanner at a 40x objective magnification. Ultimately, 300 WSI were used in training and validating the algorithm. These WSI during this phase were manually annotated for features of interest in order to teach the computer what to look for. And as Uttara mentioned, Nitin Singhal, who will speak after me, will discuss the details of this development phase. An additional 300 WSI were then used for testing the algorithm. To test the optimized algorithm, we wanted to see how well the computer's ability to detect and quantify PCM compared to trained veterinary pathologists. A panel of 5 experienced toxicologic veterinary pathologists independently evaluated a set of 300 glass slides of rat heart. This set consisted of hearts from males and females and covered a range of severities and PCM features. The pathologists were tasked to evaluate each slide and determine whether PCM was present or not, and if so, to assign a grade. The box on the right-hand side of the screen shows the grading criteria that the pathologists were instructed to use. Grades 1 through 3 were taken from a previous publication by Haley et al. And since this publication did not include criteria for grades 4 and 5, we added our own criteria for those grades, as you can see. The same 300 slides that were evaluated by the pathologists were then scanned to whole slide images and analyzed by the computer algorithm. So now on to the fun part, which is the results. So the results of the computer-based image analysis can be assessed both visually and quantitatively. So following computer analysis, whole slide images can be viewed with color-coded areas corresponding to the regions classified by the computer as a feature of PCM, with different colors corresponding to different features. So what you're seeing on the screen here is part of a whole slide image. You can see where we are in the whole section down in the right-hand corner. And at this low magnification, we can see colored areas that the computer has identified as PCM features distributed throughout the left ventricular wall and papillary muscle. And as we close up on part of the left ventricular wall, we can see that this area of the image has regions colored in red, which will correspond to fibrosis, or what the computer has identified as fibrosis, and areas in green, which correspond to what the computer has identified as cardiomyocyte degeneration or necrosis. As the color coding is removed, we can see this area contains bands of fibrosis and degenerative cardiomyocytes that correspond very well with what the computer is classifying. This is another example from a different case of an area where the computer has detected PCM features. So this is showing an earlier stage PCM change composed of mononuclear cell infiltrate, which the computer has detected in yellow, and cardiomyocyte necrosis, which the computer has detected in green. In addition to the visual output, the computer provides quantitative data as shown here. So for each whole slide image that's analyzed, the total area of the heart section is reported, along with the percentage of the heart section affected by each of the PCM parameters. So for example, in WSI number 2, in the second row, we can see that 1.27% of the heart was affected by necrosis, a little over 3% of the heart was affected by fibrosis, and less than 1% by mononuclear cell infiltrate. Summing those features, 4.5% of the heart was affected by PCM in total. And for the purposes of comparing the computer to our pathologist raters during the testing phase of the study, we used this total percentage, which I will refer to as the AI score. So just how well does the computer image analysis match with the trained pathologist for detecting PCM? So this graph helps illustrate those results. There's a lot to unpack here, so I'm going to take it one feature at a time. First, each plot point on this graph represents a slide from the test set of 300. The y-axis shows the median grade for PCM assigned to that slide by the panel of pathologists. We can see that it ranges from 0, indicating the pathologist considered the heart to have no PCM, up to 5, which is the highest severity grade possible from our grading scale. The number of cases at each median grade is shown on the right. You can note that the majority of cases were considered to have no, minimal, or mild PCM as determined by the pathologist panel. And this skew towards lower severity is typical of what we see historically for spontaneous PCM in NTP studies, as well as in the published literature. On the x-axis, we see the AI predicted score, and that's the total percent of the heart section that the computer determined was affected by PCM. Just to note that this scale is log transformed for this graph, but we can see by the trend line in the graph, as well as the statistical evaluation, that there's strong positive correlation between the grade assigned by the pathologists and the AI score. As we look at this a little bit closer, we could see at the bottom of this graph that even when the pathologist did not detect any PCM in the heart, the AI detected a low percentage of the heart to be affected by PCM features. By design, we wanted this algorithm to be highly sensitive so that we were not missing any cases of PCM. When I went back and reviewed some of the individual WSI that were in this category, I found that in these cases, the algorithm often was detecting individual cardiomyocytes with degenerative or necrotic changes or small amounts of interstitial fibrosis. So this slide is showing a couple examples from 2 of those cases. So we can see some small areas in red that the computer is detecting as fibrosis, and a few individual cardiomyocytes that the computer is detecting as degenerative or necrotic. So it could be argued whether this meets the criteria for diagnosing PCM, and indeed for these 2 cases, among the panel of pathologists, 1 or 2 diagnosed minimal PCM, while the others diagnosed no abnormality, so it may be on a threshold. But what it demonstrates is that the computer is picking up features of PCM at the individual cardiomyocyte level, or very slight increases in interstitial fibrosis, showing that high level of sensitivity. Looking at the data another way, we used a multinomial logistic regression model to look at the predicted probability of a pathologist assigned grade based on the AI score. And so what this shows is that the grades retain their order across AI scores. In other words, as the AI score increases, so does the probability that the pathologist assigned a higher grade to that slide. Again, just showing that strong correlation between what the computer and the pathologists are identifying as PCM. So to summarize, we collaborated with Ira Matrix to successfully develop a computer-based image analysis algorithm that detects rodent progressive cardiomyopathy features in rat heart sections. The algorithm results showed strong positive correlation with PCM grading performed by a panel of experienced veterinary pathologists. And this opens up the possibility for a number of future investigations utilizing this algorithm, which could include using a similar testing methodology to validate those individual PCM components like necrosis and fibrosis, looking at the consistency of the computer analysis over time versus pathologist grading over time, further investigating how AI score relates to pathologist grading and what this means in terms of biological significance. And part of this would be looking at employing this algorithm in case studies with or without test article related cardiotoxicity. Ultimately, we envision this algorithm to be employed as part of the toxicologic pathologist workflow in safety assessments, not replacing diagnosis of PCM, but providing a way to have a highly sensitive and consistent measure of PCM features, enabling the pathologist to better identify test article related cardiotoxicity. So before I hand this over, I just want to say that this work represents the combined efforts of many people. Of course, a huge thank you to the team at IRA-Matrix, who have been wonderful to work with. And in addition, a number of pathologists as well as statisticians were involved in completing this project and looking at the data, so a big thank you to them. And with that, I will let Nitin tell you more about the algorithm development.
Thank you, Deb. Hello, everyone. Before jumping into the algorithm details, I would like to give a brief about the research activity at IRA-Matrix. We at IRA-Matrix are constantly pushing the boundaries of the state of art in artificial intelligence with a special focus on computer vision. AI research in IRA-Matrix is practiced as a multidisciplinary exercise where insights from data-driven machine learning and deep learning is fused with domain-specific knowledge to amplify the quality as well as the causal accuracy of the predictions which are derived. We are creating state-of-the-art perception and reasoning capabilities for our AI technology to observe and understand contextual meaning. Our capabilities in computer vision, machine learning, deep learning, knowledge representation, reasoning are used to robustly predict required information. Some key challenges we tackle include a lack of sufficient labels needed for traditional supervised learning approaches, the need to ingest and link multiple data modalities, efficient deployment of complex deep learning models, and the need to build AI solutions that are interpretable. We are developing learning systems for few-shot, zero-shot, class imbalance kind of problems that learn from minimal labels to predict behavior. We are developing virtual staining systems that can produce virtual immunostains images from existing H&E whole slide images. To overcome some of the challenges in deployment, we are developing knowledge distillation techniques, which effectively learns a small student model from a large teacher model and compresses the whole network architecture. Our researchers and engineers are constantly aiming to apply these state-of-the-art solutions in different aspects in life sciences. For example, improving the disease diagnosis, augmenting pathologists and physicians by increasing workflow efficiency to save precious time and the burnout. Getting back to cardiomyopathy and the process flow, the figure shows the main step that constitutes the algorithm pipeline. Some key elements of the algorithm includes a multi-resolution ensemble of segmentation models to delineate normal as well as abnormal parameters, pre-trained model weights for identifying normal parameters such as RBC and blood vessels. These normal parameters are used to eliminate false positives identified in the prediction of abnormal parameters. An alert score is assigned to a whole slide image based on total percentage area of the affected tissue. In the data, we use the data in this work consists of a set of whole slide images. Our training data includes 270 and validation data set comprised of 30 whole slide images. A team of expert pathologists provided their review of the images in terms of pixel level annotations. These annotations were marked to identify pixels and regions belonging to fibrosis, necrosis, MNC, and mineralization. During the pre-processing step, each of these whole slide image was divided into tiles. A table on the left shows the magnification and the number of tiles used while training as well as validation. As you can see, the magnification is decided based on the size, context, and appearance of different parameters in the whole slide image. We also used a testing dataset comprising of 300 images to evaluate our developed algorithm. Getting into the network, we present a customized deep learning network to improve upon the segmentation performance of a fully convolution network. The proposed network is a refined UNet architecture with a ResNet 50 convolution backbone, as illustrated in this figure. The network consists of skip connections that refine the semantics and the spatial precision of the output. A refinement is introduced in the encoder part, where we replace the heavy VGG module with the ResNet 50. The residual connections in ResNet 50 helps in avoiding the vanishing gradient problem and also makes the training more predictable with faster convergence rate. Further, the reduction in training parameter improves our training and the inference time as well. The table summarizes DICE performance achieved using our algorithm pipeline for each of the parameter. The DICE similarity coefficient, also known as DICE coefficient, is a statistical tool which measures the similarity between two sets of data. Here, two sets of data are ground truth label marked by expert pathologist and the predictions by the algorithm. As shown in the table, the algorithm achieves significant overlap with the ground truth label. From a qualitative analysis, the figure shows an input image as visualized in our IRA matrix viewer. And here is the output image. We can show the output shows qualitative performance of our algorithm and percentage area of each of the abnormal parameter. Besides the whole slide image score and quantification, the system also outputs pixel level overlays, as you can see in different colors, red being fibrosis, green showing the necrosis, and the yellow showing the MNC. These overlays can be used by pathologists as a second read to confirm that no area from the image is missed from analysis, in addition to improving the accuracy and precision of severity scoring. Here is another example of an input image and the corresponding output. With this, I open the floor for questions.
Right, Nitin, I see several questions in the Q&A box here. To begin with, there is a question which goes, I think, Deb, this one is for you. Based on the training of the AI algorithm using 300 slides, is this ready to assess PCM independently without any supervision by a pathologist? Deb, could you address this one, please?
Yeah, so I think we would like to do a little bit further kind of testing of this in a real life situation, if you will. Applying this, I'd like to see it applied in a study where either artificially or based on a real study, we have treatment related or group related differences in PCM severities to see if applying the algorithm reflects that and highlights that difference. And sort of as an aside to that, I think that the way that we would envision this algorithm being used is not necessarily, completely independent of a pathologist overseeing it or overseeing the use. I think it would be combined along with the pathologist viewing the output as well as what the color-coded images and getting a feel for what the computer's picking up in the hearts. So having it be part of the pathologist workflow. I don't think we're quite there yet, but close.
Right, thanks, Deb. Nitin, there is a question for you. It says, could you comment on the intersection over union parameters? Have you published the results of your study or a code or data set anywhere?
Yeah, thank you, Uttara. So we have evaluated our algorithm segmentation performance in terms of dice as well as intersection of union. So dice performance I already showed, and similarly on the intersection over union performance, we got around between 80 to 90% accuracy. And from a publication point of view, we have published one of the paper in toxicology journal, but not any particular paper in a technical conference.
Right. Thanks, Nitin. Right. There is another question, Nitin, here, which is about how have you taken care of artifacts like stain quality, old slides, etc.
Yeah. So in pre-processing step, as part of our implementation, we have algorithms for detecting artifacts, something like blur, fold, and these artifacts. So there are separate algorithms which are used in the pre-processing step to remove some of these artifacts, and then we go about training and testing our algorithm.
Right. And there's another question here which asks, does this algorithm run on whole slide images or only on a region of interest?
It runs on both things, so it can run on a whole slide image as well. And if the user marks ROI, region of interest, then the algorithm can run on the region of interest as well. So our software, which is our viewer, provides both the option.
Right. I think there is one more question which is asking about the DICE score and how did you look at the accuracy of segmentation. I think you answered that in one of your slides, but would you want to?
Yeah, so we have measured segmentation performance in terms of DICE and intersection over union. DICE is compared with the ground truth annotation marked by the pathologist and what the algorithm predicted.
Right. Right. Deb, there is one for you. Is there a difference in fibrosis scoring between trichrome special stains versus H&E stain? Or Nitin, maybe you could take that because we've done fibrosis scoring on Masson trichrome stain as well as picroserious red. So the question is whether there is any difference between scoring between H&E and the other special stains. Yeah, one of you. Deb, are you taking that one?
I can just comment on maybe the visual aspect from the pathologist point of view. I do think looking at a special stain like trichrome certainly highlights fibrosis more than just on an H&E section, and so that might have some influence on a pathologist's eye picking up the amount of fibrosis and could change their grading just from a visual aspect. But maybe Nitin can comment on that from more of a computer aspect.
Yeah, so from an algorithm point of view, we can develop, basically, we have experience working on different spatial stains. This algorithm was developed using H&E stains, but a similar approach could be taken to, which can be applied on spatial stains as well.
Right. Deb, this one is for you. How long did the study take?
So I think in total, we kind of started this initiative in January or February of this year and had the results I presented here by July. So about 6 months or so.
Sure, that's great. Yeah, there are several more questions also. Okay. There we go. How was training done, especially classes like fibrosis? Was it annotated before patch extraction or after patch extraction? Nitin, I'll take that.
Yeah, I'll take that. Annotations were done on whole slide images. We used our marking tool for making these annotations. The pathologists use this tool to mark annotation, and then the patch extraction was followed.
Right. I guess with this, we've answered all of the questions. We've addressed all of the questions. So thank you once again to all of you for joining us today. And if you have additional questions or any feedback, you can always write to us for queries either on this solution or any of our other services. We look forward to interacting with you in our forthcoming webinars on AI applications in reproductive toxicology as well. Meanwhile, do take care of yourself and stay safe. Goodbye and have a great day.
Thank you.